here in this figure, describing these motifs from native jelly rolls, what they look like, and describing basically the phi-psi angles that are represented in the Ramachandran plot across the different 5 mers, 9 mers that you would find with this specific motif. And so that was part of defining this motif, right? Is okay, if we are starting from that space, let's look at what nature has done and then see if we can develop rules to generalize across the topology so we can make our own brand new jelly rolls. And so here you actually see those beta arcades that I was talking about, enumerated and forming, and then down to the literal mathematics of it, like it's a specific angle that actually forms this, and that angle that can be translated to a vector in coding space, right? And that vector mathematically can be enumerated across multiple different combinations combinatorially to make multiple backbones. Those backbones, we then, this is the forward folding funnel I was talking about. We do a sequence optimization. I think here we were still using fast design, which included layer design, I think, which basically highlighted, okay, surface residues need to be polar, core residues need to be hydrophobic, and then you had some boundary residues that are a mixture of each one. And you were scoring based again on Rosetta energy. You stick for delta G of folding, and so that's what you have on the y-axis. Axis is RMSD, which is basically your structures here in green sampled, and then all the 20,000 plus simulations that you have done of the fragment assembly ranked by RMSD and Rosetta fold of energy. And a successful funnel, which is what we call the forward folding funnel, is one that pointed downwards like this. You had some simulations that approached in RMSD, so that means they were very close in resembling your designs, but you also had some that were low in energy, because you see majority of these are high energy structures, which means they are likely not going to fold into the sequence, is likely not going to fold into this conformation. But some of these low energy structures, likely the sequence is predicted to fold into the actual structure. And now, because it's close in RMSD to your design, so it's actually the structure that you have designed for. The first graph here is something called circular diacharism. We're actually going to do this for our protein, you guys. So this is an experimental characterization. I did this. This is my plot. Actually, this is my graph I'm showing here. And so circular diacharism essentially measures what's called elicity, whatever. So this is like the chiralness of your protein, right? And so essentially it describes secondary structure elements of your protein. So a beta sheet type protein will have a specific signature that's different from a helix type protein, that's different from a random coil protein.